My name is Fumilola Favor Hanjari, and for this year's graduate research conference, I'm presenting on pancreatic cancer targeting using a phage display-derived peptide MCA1. My research mentor is Dr. Mitchell Soendegard in the Department of Chemistry. I'm a second semester chemistry graduate student. Okay, pancreatic cancer accounts for about 3% of all cancers in the United States. And in total of all cancers, it accounts for 7%. Globally, it is known that this statistics keeps increasing. And this is due to the fact that when it comes to pancreatic cancer, it's almost too late sometimes. I recently had a conversation with someone whose friend died of pancreatic cancer, and he piqued interest in my research and was like, oh, I'm interested in finding out what the outcome of your research would be, because for my friend, it was almost at the terminal stage and nothing could be done. So this is due to the fact that there is aggressive metastatic progression as well as the asymptomatic nature of the disease. So finding a way around detecting early diagnosis of it will lead to overall treatment and improve the overall survival rate of patients with pancreatic cancer. Okay, so bacteriophage display technology, known as phage display technology for short, is it uses libraries that contains millions and trillions of different genetically modified virions to express foreign peptides. And so previously, a phage display selection of peptides, we have a phage library in our lab. So a phage display selection of peptides that specifically bind to pancreatic cancer, which is MIA-PKA-2, has been done, which led to the discovery of the peptide MCA1 that binds to MIA-PKA-2. So here in this research of mine, I plan to establish the cell line specificity of MCA1 to pancreatic cancer cell. Is it binding specifically to pancreatic cancer cell, which is the MIA-PKA-2 cells, or is there an attraction to other cancer cells? So I'll be testing it with ductal adenocarcinoma, CFPAC1 cells, pancreatic adenocarcinoma epithelia cells, the Panc 10.05, and then the human pancreatic adenocarcinoma cells, the HPAF-II, will be tested. And this is in order to establish the specificity specifically to MIA-PKA, or is it binding to other cells? So to achieve this, modified ELISA protocol and fluorescent microscopy will be used. And then here is a brief description of what an ELISA procedure looks like, even though it's going to be plated not here, but on a 96 well ELISA plate. So, but before then, we grow the cells. The cells are frozen. So we grow the cells in medium. So cells and mediums are purchased from ATCC. This is what I'm trying to describe here. This is what it looks like under the microscope when cells are grown. It shows a highly dense environment for MIA-PKA. This shows a sparse density growing, and then this is a highly populated. This is a growth going on maybe day 1 or day 2. They grow so fast. MIA-PKA cells grow so fast. So to grow the medium, they are being propagated in DMEM for MIA-PKAs. Each of the cells have their corresponding medium. So supplemented with 10% LBS, 5% all serum, that's for DMEM medium. And then for pancreatic adenocarcinoma cells, it will be propagated in RPMI 1640 medium, supplemented with 10% human recombinant insulin and 15% FBS. Then human pancreatic adenocarcinoma, which is the HPAF-II cells, will be propagated with the EGUS medium that is supplemented with 10% FBS. In our lab, we usually had gentamicin as an antibiotic function for the growth medium. Okay, so once the cells are grown, ELISA can then be performed. So what happens in ELISA is that a baltinated peptide will be incubated with the MIA-PKA cells to determine the binding affinity. So this is the cell. Inside this epithelium shown here are the cells. The cells are present here embedded in the cell medium, and then they will be probed with HPAF-II conjugated spectravine and measured spectrophotomically. Once plated on the 96 well plate, procedure of ELISA, which I will go over very soon, will be done on them, and then they will be measured spectrophotomically at 405 nanometer after addition of the HPAF-II peptide. So it's a major thing in this ELISA procedure. The HPAF-II is a major in the ELISA procedure because it's supposed to bind to it. So measurement to be done with the spectral mass 50 micropace. So for the ELISA procedure, the binding and cell specificity of MIA-PACA-2, I plan to establish the specificity to MIA-PACA-2 and not to other cells, is to perform an ELISA procedure where the cells will be incubated with biotin and DMSO acting as a control vehicle for 1 hour at 37 degrees centigrade and 5% CO2. The cells are then washed with 1% bovine serum albumin in PBS and fixed with 10% formalin for 10 minutes. So the cells are washed extensively after the formalin addition and incubation at room temperature for with 1% BSA and PBS and blocked using 10% FBS glycine and 0.05% 20 in PBS for 30 minutes. So the bound peptides will then be probed. This is where the HRP comes in. It functions as a probe, will then be probed by oral dish peroxidase conjugated streptavidin for 1 hour, incubation for 1 hour inside the 96 well plate at room temperature, after which the cells are then washed with 0.05, we call it the second wash, 0.05% 2020 in PBS. Bound peptides will be detected spectrophotomically at 405 nanometer after the addition of the HRP substrate, and this will then be used to determine the EC50. For those not familiar with the term, EC50 is the half maximum effective concentration, and it refers to the concentration of a drug which is able to cause like a halfway response between the baseline and the maximum after the exposure time to the drug. So it will be used to determine the EC50 of the procedure. So this is an example of a dose response curve. So this is a data from a previously done research in our lab. So showing a dose response study of biotin, MCA1 and MCA2 binding to MIA-PACA-2. They both bind specifically well to MIA-PACA-2, So for this research, it was tested with other cell lines, which are listed here, of 3, the ovarian cancer cells, the NCAP cells, the HTER cells. So, but I am doing for other cells, we need to test it more and more to be sure that it actually has real specificity to MIA-PACA cells and MIA-PACA-2 cells, and not to a variety of other cancer cells. So the whole purpose of the research is that elucidating the pancreatic cancer binding specificity of MCA1 may be a way forward in the development of peptide as a diagnostic method for pancreatic cancer. Thank you so much, and kindly drop your questions and then comments on this presentation. Thank you so much. Once again, my name is Fumilola Favor Anjari.